So thank you everyone for joining us today for today's session of the public webinar series. Today, I am going to start off by sharing some updates, and then we'll go into our featured speakers. If you are unable to watch or attend any of our other webinars, please feel free to go to our YouTube page, as you can see here, which has all of our previous webinars recorded for you to watch at your leisure. If you have any feedback, questions, or would like to be added to our listserv, please email us at help@cfdeknowledge.org. Now I'm going to go through some of the events that we are going to be attending through the rest of 2025. And as we have previously, this year we are going to be at the American Society of Human Genetics in Boston, Massachusetts. And I want to give some information about the various events that we'll be hosting throughout the week. First, we'd like to invite you to mix, mingle, and map the Common Fund data ecosystem. This is going to be Tuesday, October 14th from 7 to 9 p.m. at the Westin Boston Seaport District. This event is going to focus around groundbreaking research, and you'll also have the opportunity to connect with fellow scientists. We'll have cocktails, appetizers, and there'll be posters for people to share anything about their research or any scientific updates that they'd like to share. We have a wonderful group of people that are going to be in attendance. As you can see on the right, these are some of the organizations that will be joining us. This event is free. You do not have to be registered for ASHG to attend. But as you can see, we have a registration link here. So if you're looking to participate, please register just so we can see who will be in attendance. And if you're interested in learning about how to present a poster at this session, all the details are here. With that, we will also have our exhibit booth, which will be 1428. And this will be Wednesday, October 15th through Friday, October 17th. And this is your opportunity to meet with us from members of the Common Fund data ecosystem, to give updates from our science, explore tools through personalized demonstrations. We'll have the different leadership through our ecosystem, and we'll also have some spotlight hours if you want to speak with individual DCCs. We're very excited about this, and we can't wait to meet all of you in person. We'll also be hosting an ancillary session again this year, and this is going to be revolved around investigating mechanisms underlying disease with Common Fund data ecosystem data and resources. This will be October 16th from 11:45 a.m. to 1:15 p.m. In the convention center, and we really look forward to seeing you all at this as well. If you want to look more at the various events and details, we have this website, which is available to you to see the calendar events and additional details for what I had just explained about before. One other thing that I wanted to share is the training system has the Decoding the Data Ecosystem podcast. This is more than just a podcast. It is an opportunity for you to learn more about the ecosystem, the different science that goes onto it, and the researchers' experience throughout. So Dr. Alyssa Dillman from the Training Center is the host, and there are 9 episodes that you can listen to available now. So with that being said, I'm going to hand it over to our featured speakers. And Bernard, you can go ahead and get started when you're ready.
Thank you so much, Mackenzie. Can you see my screen?
Yeah, thank you.
Right, let me maximize the slide. So hello everyone. My name is Bernard Bono, and I'll be talking as well co-presenting with Peter Hunter today on the transport processes in multiscale physiology. So I would start by saying that this discussion will feel relatively brief because comparatively there's so much to say. And I also hasten to add that this is really a presentation of works in progress that Peter and I are carrying out in extending the core use case for SPARC, which is a Common Fund project that focuses on the autonomic nervous system. And we are extending its core use case to now also cover the requirements for the detailed multiscale modeling of transport processes, with particular emphasis on the absorption, distribution, metabolism, and excretion of drugs. I would also point out that this requirement was developed over the past 24 months, thanks to the efforts of the Common Fund Data Ecosystems COMPA effort that put us in touch with an amazing community of pharmacology modelers that are mainly focused on maternal and pediatric health. And with them, we developed means by which we have now a roadmap to extend the infrastructure that SPARC has developed for autonomic physiology to now link it with pharmacology modeling across multiple scales. And the core requirement here is really to ensure that when we try to model transport processes over multiscale tissue connectivity, this must really be founded on an explicit representation of biophysical knowledge. So not only an explicit representation of energy transforms that are driving transport processes, but also to ensure that the representation of tissue compartments are themselves anchored into biophysical reality. So the talk today will be in two parts. I will first take you through our approach within SPARC to represent tissue connectivity. And for this, I will introduce you to the appenatomy formalism that relates to networks of compartmental connectivity. And then this will segue by Peter taking us through the bond graph formalism, which is an approach to represent biological processes as a series of energy transforms that will be then implemented as a network of ordinary differential equations that are overlaid onto the appenatomy representation of compartmental networks. So really, the take-home message here is that to have an explicit representation of multiscale transport that is amenable to modeling and simulation, you need to combine knowledge graphs for tissue conduit networks, and that's the appenatomy formalism, with knowledge graphs for biophysical networks of ordinary differential equations that are explicitly representing energy transforms, and that's the bond graph formalism. But let's not rush. Let's start with the background. Both Peter and I are driving investigation for the SPARC Common Fund project. And SPARC, by the way, I would invite you to visit the SPARC portal at sparc.science, where we have some rather interesting resources for autonomic nervous system physiology. And SPARC is driven by the need in particular to support the development of so-called electroceuticals. Electroceuticals are essentially electronic cuffs that are placed around peripheral nerves to be able to modulate primarily autonomic outflow, but also in certain cases, interoception. And we'll find that really the tissue basis for SPARC representation is rooted in the autonomic nervous system, which is a primarily motor outflow of the brainstem in particular to the various visceral organs, but also to deal with the way that the body is collecting feedback from these visceral organs, the so-called interoception. And many of the neurons relaying this information are also hitching rides along nerves that are primarily associated with autonomic outflow. But really, the autonomic nervous system teaches us a lot in terms of the commonality by which the body handles visceral sensation and visceral parameters. And in particular, that the autonomic nervous system tweaks our physiology in one of two ways. Either by influencing smooth muscle contraction, I'm talking in very generic terms here, of course, either by tweaking smooth muscle contraction to affect pressure or resistance, or else by driving or modulating secretion. So essentially, it is a form of pumping of substances into one of the major thoroughfares of the body. And in that sense, if one were to create a kind of generic representation of autonomic physiology in general and transport physiology in particular, one needs to develop a notion of the key thoroughfares of the body, the key fluid thoroughfares of the body. And in fact, there aren't that many. It's quite a finite amount. So you have autonomic neurons that will both track the parameters, but also influence contractility of smooth muscle around blood vessels, so primarily by influencing total peripheral resistance. So that's one thorougher, the blood thorougher. You have bile thorough, the outflow from the liver into the gut, primarily through the contraction of the gallbladder. The urinary thorough, starting from nephrons all the way down to the bladder. And of course, around the calyceal system, the ureter and the bladder, you have smooth muscle regulating there. The airways, where you have sympathetic outflow that is modulating the caliber of our major conducting airways, but also a lot of sensing going on in terms of stretch. The gastrointestinal tract has its own, as it were, primordial brain of plexuses around the smooth muscle to regulate the peristalsis of chyle, which is digested food, but also it's regulating the secretion of glands within the wall of the gut itself. CSF is yet another major thoroughfare of fluid in the body, and its flow is mainly regulated by regulating secretion of CSF through the choroid plexus. And finally, pancreatic juice, which is now an overtly secretory organ for the digestion of our foodstuffs. So these are essentially the major fluid thoroughfares in the body that need to be explicitly represented, because they are crucial for general physiology representation in the first place, but also extremely relevant in the absorption, distribution, metabolism, and excretion of drugs. So this is the sort of long-range thoroughfares that we need to have explicit representation of. But then we also must be mindful that the autonomic nervous system is tweaking the exchange between these thoroughfares. A lot of physiology is happening by regulating exchange between the major thoroughfares in the body. For example, in the lung, we have the exchange between the air thoroughfare, which is represented here by an alveolus, but of course this air is linked to the outside via the conducting airways. And of course the capillary network around the alveoli. So the exchange between the air thoroughfare and the blood thoroughfare. In the liver, we have exchange across three main compartments of fluid. Bile with lymph, so we have bile in green here and the space of this, which is a thoroughfare of lymph. And then between the lymph and the space of this and the blood within the cords of the sinusoids of the liver. In the gut, again, we have regulation between exchange between chyle and the lumen of the gut, blood and lymph. But you can see these patterns re-emerging. So these are recurring motifs that are relevant for transport physiology. Between urine and blood, so between the vasa recta, for example, and the loop of Henle in the kidney. And uniquely to the placenta, between a blood thoroughfare of one organism, that is the mother, and the blood thoroughfare of a second organism, which is the fetus. But nevertheless, the same biophysical constraints are acting over all these interfaces, in the gut, in the lung, in the brain, in the placenta, in the kidney. So we have, therefore, a kind of underlying theory of exchange that can be now represented as a knowledge representation that can be used to organize our knowledge about these crucial processes in the body. So this is where, this is provided the motivation to develop a toolkit, which we refer to as app anatomy, to provide us with the means to assemble in a fair and well-organized way multiscale conduits and to represent fluid flow across multiple scales, both along the major thoroughfares that we talked about, but also across them. So between, say, blood and urine, or blood and chyle, etc. And the origin of this work goes back all the way to 2013, where Peter and I started thinking in terms of what are the key biophysical constraints that define a basic tissue domain. What do we mean by a basic tissue domain? Essentially, we were thinking in terms of, we do have a pretty thorough understanding of a protein domain, right? A protein domain is an independently folding unit that has a backbone and has side chains and essentially the information encoded in the primary structure of that polypeptide chain informs the folding and therefore the 3D architecture of a protein domain. What is the equivalent? Is there a way for us to think in terms of a tissue domain that satisfies the biophysical constraints for cells at that particular scale level? And the best way to think about it is to think of a cell almost akin to a side chain, an amino acid side chain. And the cell is secreting factors on a paracrine basis. And the cell needs to coordinate with other cells within diffusion distance through the sensing of factors secreted by other cells. So essentially, the first point, the first constraint for a tissue domain is that cells within the same tissue domain need to be within diffusion distance of one another. But the second constraint is that these cells need to survive and need to communicate with cells that are beyond the diffusion limit itself. So cells need to be also within diffusion distance of a central duct, a central conduit that is one of the major fluid thoroughfares that we have just described. So the central tube that you see here could be a blood conduit, could be a urinary conduit, could be a bile conduit, but essentially it needs to be a conduit for the long-range thoroughfares of the body that we've just described. So here are the two constraints acting together. A tissue domain would take a cylindrical form where there is a central conduit for a major thoroughfare that is within the diffusion distance of a cuff, a peripheral cuff of cells, such that any two cells within the same cuff must be within diffusion distance of each other. And it is the basis of what we refer to as the functional tissue unit, which is a term that we coined back in 2013 to underpin a tissue domain that is satisfying very, very explicit biophysical constraints. And essentially, there is a very distinct topology to this tissue domain. That is, there is an asymmetric rotational unit with an axis of rotation. The innermost layer is a long-range fluid thoroughfare, and there's a cuff around it of parenchyma that is within diffusion distance of the thoroughfare at the center. And it is on the basis that we develop then a generalization over the representation of fluid conduits, in which, in fact, this functional tissue unit is but a special case of a generic representation of conduits in the body. Which means that when you look at any large-scale conduit, any thoroughfare in the body, whether it's the airway thoroughfare on the left and the urinary thoroughfare on the right, they do consist at the termini, where you have tubes that are narrow enough to permit diffusion. They do consist of functional tissue units, but then you also need to have larger caliber conduits that permit mass flow. And these larger caliber conduits have a wall that consists of FTUs around blood, such that blood can provide the necessary building blocks and remove wastes to maintain the integrity of the large-scale conduits. So essentially, you can start seeing the sort of rationale across different tissue types by which we build representations of tissue as a network of compartments in terms of FTUs that permit sideways diffusion. But themselves, these FTUs are themselves connected to concatenations of larger bore, larger radius conduits that permit mass flow and have the appropriate compliance and material properties that for the larger vessels that permit the pressures needed for this large scale flow. So in that sense, apenatomy is nothing but a knowledge representation to represent conduits. And these conduits could be FTUs at a small scale, but could also be large segments of vessels from the aorta, from the trachea, etc. But these conduits have a very stereotypical underlying knowledge representation. There needs to be an axis of rotation. There needs to be layers that border one another. There needs to be polarity because direction matters. And they need to be associated with metadata, for example, distributions of thickness and length, and of course, be annotated fairly by ontology terms that are coming from the Open Biomedical Ontology Network to ensure that the knowledge that is represented there is explicit and shareable and findable by the rest of the community who are using the same standards. And then, of course, bear in mind that conduits have a limited but very distinct set of topological configurations. That is, a conduit can be either, from a conformational perspective, be a tube, that is a cylinder that's open at both ends, a bag, essentially a cylinder that's closed on one end, or a cyst, essentially a cylinder that's closed at both ends. And using this very limited set of constraints, of rules, we can use this as a kind of assembly toolkit, as a kind of Minecraft toolkit to build any type of thoroughfare that we want, but also not only the thoroughfare themselves, but also the interaction between one thoroughfare and another, as we shall see. And essentially here I'm giving some examples of a topological tube, bag, or cyst in terms of biological examples. So a tube could be a membrane channel or a segment of proximal tubule. A bag could be the Bowman's capsule at the end of the nephron. A cyst could be, of course, a cell, but also the whole of the peritoneum, or simply a vesicle as a subcellular compartment. So how do we use these assembly rules to build connectivity for tissues using the app anatomy formalism? Well, we develop rules whereby we can concatenate conduits to one another. And essentially what you have here is a representation of the entirety of the urinary tree, starting with the urethra on one end as a layered representation of concentric tissues. The urinary bladder is here, but then you can see the caliber itself, although not apparent in the topological representation, the caliber of the canonical tube starts going down until you reach the nephron, where you start then meeting the actual FTUs, the functional tissue units, across which lateral diffusion is permitted. But you can use this approach to represent the whole of the urinary tree, or the airways, or the biliary tree, etc. And I'll give a couple of examples here. So this is a first assembly of the anatomy concatenations to represent the whole body in terms of a homunculus that represents flow across multiple scales. So you can see here that on the left, you can see sort of the body as we know it. And on the right is the homunculus itself. This is what's referred to as a two-map. The T in the middle representing flow for CSF, cerebrospinal fluid. The innermost O represents circulation of blood, so the endothelial tubing. And the outermost O in green is representing epithelial flow. And then you can see these tubings here that represent different types of arborizations, concatenations. So here you have conduits for the lung, the trachea, going down to the conducting airways, reaching the alveoli at the end, the very tip here. So the FTUs will be at the tip of these tubes. But each tube is made from a concatenation of these apenatomy conduits. So if we take an arbitrary conduit, say the major calyx and the kidney, then we can open it up and see the layered representation of this particular conduit. And each layer is associated with material properties that map to the ontologies that we just discussed earlier. And this is just one conduit, but in fact, it is linked as a concatenation to the entire set of conduits that make up the epithelial nephron. But then we will see how we can use this representation then to connect thoroughfares from different fluid types. Where was I? So I'll skip that. Oh, yes, and perhaps I'll also give you a very brief demonstration of the underlying editing suite that we use to build these concatenations. So this is over the apenatomy editor itself. Oops, sorry about that. I shouldn't have done that. So here you have the two-map again. So the outermost circle is the epithelial conduits. The innermost arrangement here is representing the circulatory system, blood. So here you have the mitral valve going into the aorta. So these are the major blood thoroughfares for the systemic arterial tree. On the other side, you have the major blood thoroughfare for the systemic venous tree. And then in the middle here are the blood vessels, the thoroughfares that are going through a particular organ, in this case, the kidney. And this is where the blood FTUs for the kidney exist, because this is where sideways diffusion happens, okay? But these are just the sort of long-range concatenations for long-range flow. But where do we take it from there to now represent exchange between fluid thoroughfares? So essentially, we can think of the concatenation, the assembly of short-range coalescence in terms of sideways diffusion between these FTUs. So essentially think of FTUs as layered structures, which, when fused by sharing the outermost layer, create a coalescence, which is essentially a stack of compartments that really track the exchange between one fluid thoroughfare and another. For example, here we have a sideways coalescence between a segment of proximal tubule, which is an epithelial tube in the kidney. So the central fluid here is urine. And on the other side, you have a capillary from the peritubular network in the kidney that is really centered around the blood thoroughfare. But when you open up these conduits, you get a 5-layered representation, which is the order, the sequence of compartments that have to be crossed to really account for the exchange between blood and urine in the nephron. And I think I also have a visual representation here in 3D. So here we are in the kidney. We're zooming in towards the peritubular capillary for the proximal convoluted tubule here. So you can see the two appenatomy cuffs. I will also overlay an appenatomy representation of an epithelial cell in green on the right, and then an endothelial cell on the left. And then we open them up. And here's the same 5-layered representation that I just showed you in the previous slide. So here you have the layer of urine. So this is the urinary thoroughfare. In green, the representation of the epithelium. In gray, you have the basement membrane. In pink, the endothelial layer. And in red, the thoroughfare for blood. But also bear in mind that using the same formalism, we can also open up the cell, say the endothelial cell and the epithelial cell, and they themselves are represented in using the same appenatomy formalism. So here we have the luminal surface of the endothelial cell that is in fact embedded in the blood compartment. Here we have the abluminal surface of the endothelial cell that is embedded in the basement membrane. And here within the endothelial layer itself, I place a tube that really links the luminal surface to the abluminal bags of the appenatomy cell representation as a chain, as a concatenation. But what is the structure here? Well, this would be a membrane channel, and the membrane channel itself is again represented using the same appenatomy formalism. And there's a cytosolic component to the membrane channel, a transmembrane component to the membrane channel, and an extracellular component to the membrane channel. And we map the sequence ranges using UniProt to be able to see which subsegment of a polypeptide chain is in which compartment. That is now well defined using the appenatomy formalism for these compartmental networks. And yes, I forgot to also show this. Well, I guess this video is a bit redundant, but again, this uses the same sort of visualization to remind us that essentially here we have two tissue domains that are interacting with one another in the same way as you have the quaternary structure in a polypeptide assembly to create protein complexes, right? But in this case, we have two backbones, which are two major thoroughfares, and the side chains are the cells that are interacting with one another. And with that, now, as Peter will soon show us when he takes over the second half, we are using this essentially well representation of compartmental networks, particularly in terms of these coalescences that track the compartmental sequence between one thorifer and another, now to overlay onto this sort of layered representation of compartments, networks of ordinary differential equations that are based upon that are derived from the bond graph formalism that tracks the energy potentials associated with, in this case, molecular concentrations, and then tracking the fluxes between one energy node and another that really tracks the rates of flow associated with these molecules. Essentially, just to remind you that we are working with a large community of pharma modelers who are coming from the Maternal and Pediatric Precision Therapeutics Hub, MPRINT. I would also strongly invite you to visit their website, mprint.org. And with this community, we're really trying to map the compartmental representation and the bond graph representation that we are discussing in this seminar to really address the representation of physiology-based pharmacokinetics. And this is what I'm presenting here is really born out of a common fund data ecosystem workshop that Peter and I held in collaboration with the MPRINT community, really to discuss two-way exchange of skills as well as requirements between the pharmacology modeler community and the common fund data ecosystem. Essentially, what we're driving towards is to leverage upon the amazing resource that is the Pharm GKB, or now I guess it's called Clin PG, which is really a resource that deals with the handling of drugs by the body, but also on the pharmacodynamics, that is the effect of drugs on the body. And we're really leveraging a crucial resource that they have created over many years, which is networks of gene products that are relevant to the absorption, distribution, and metabolism and excretion of drugs. And we want to be able to take these hand-drawn diagrams and map the gene products and therefore associated with the appropriate external links for Pharm GKB pathways, UniProt IDs, as well as gene IDs, as well as linking them with the IDs from Chembl and PubChem to small molecules that are relevant to the pharmacology scenarios that we are seeing here. And what we're doing is we are now in the process of mapping the small molecules and the gene products relevant to pharmacokinetics onto the tissue compartmental network in app anatomy, and then linking mathematical models of flow of transporters of pumps that are relevant to these drugs onto the same tissue network to be able to provide in the long term a service infrastructure for pharmacology modelers to leverage common fund data ecosystem resources in support for their modeling objectives. With that, I think I will stop here and pass on to Peter to take us through now how we represent networks of energy transforms in the context of bond graph formalisms that link back to the network of tissue connectivity that I've just discussed. Thank you so much. Over to you, Peter.
Okay, thanks, Bernard. I'll share my screen. So I want to follow up to Bernard's talk by talking about the way we handle the physics in multiscale processes through a framework called bond graphs, which I'll try and explain. And we start with the units shown on the left, which are the fundamental units that we'll use for defining all flows and potentials in the bond graph framework. And we've really got three primary conservation laws that we need to satisfy at all scales and we're modeling processes in the body. And so the key concept here is energy, which is the capacity to do work. Work is the motion against an opposing force. And of course, we've got to understand the generation of heat from those processes, the so-called inefficiency of those processes. And by the way, a lot of the thermodynamic side, of course, comes from Gibbs, as shown here. So the key thing with energy is you can transmit it, store it, and convert it. And when you're converting it, you're converting energy between only 4 different forms. It's either mechanical energy, it's electromagnetic energy, it's chemical energy, or it's heat, nothing else. And so we've got to be able to capture, the Von Graph framework will capture the mathematics of being able to convert energy between those forms and deal with the transmission, storage, and conversion. So if you think first of all about transmission, and let's take mechanical first. In the case of mechanical energy, there's some movement which could be measured in meters per second being driven by some potential, a force, a joules per meter, which is a Newton. Or if it's a fluid, then it'll be a volume flow in meters cubed per second driven by a pressure or energy density in joules per meters cubed, which is a Pascal. If it's electrical, you're driving a current flow, which is coulombs per second, with a potential, which is joules per coulomb or volts. If it's a chemical potential, you're driving a molar flux. And all of these fluxes are the ones that Bernard's referring to in the conduits. And in the case of a chemical potential, you're driving it with joules per mole. In the case of heat, you're essentially moving entropy. The flux is entropy per second, and you're driving it with joules per unit entropy, which is Kelvin, degrees Kelvin. So you can think of the potential as the push that's pushing along the quantity given by those units, and its rate of change is the flux. And so the product of the potential with the flux is always power, because you've got joules per unit multiplied by that unit per second. So that you end up with joules per second or watts. So what we're doing then is representing energy, and we've got to give an arrow head to indicate a positive direction. But the orientation of the arrow is not significant. We're just saying there's a flux of energy. And what we've got to do is to, on each of those so-called bonds where we're transmitting energy, we've got to represent both the flux V in green, which is the rate of change of a quantity, and the potential that's driving that flux U. The real insight into how Von Graphs works came originally from Henry Painter at MIT, who was working in departments of mechanical, electrical, chemical engineering at MIT, wanted to be able to talk across disciplines with his colleagues in all departments, and realized that, in fact, if you take an energy approach, and so, for example, if you're summing up the product of that potential and flow, which is an energy per joules per second, and you're summing up, in this case, for 5 transmission lines for that energy flux, then the sum of those must be 0 because you're conserving power. But if you then say, let's consider the case where the potential in each case is common to all 5, and we'll pull it out and put it into the middle here, which Henry Painter called a 0 node, then that means you can take the U out the front of that summation because it's common to all the bonds, and you're left with the sum of V equals 0, sum of fluxes is 0, which is just conservation of mass or charge. Alternatively, you can imagine a situation where your transmission lines of energy are now have all the same value of V, the flux, but have got different potentials, and Painter called that a 1 node. And now when you take the V outside the summation, you're left with the sum of U's equals 0, and that's conservation of energy. So in fact, by applying conservation of power appropriately under these different conditions of either a common potential or a common flux, you've satisfied, and you make sure you're satisfying power conservation, then you're satisfying all the equations of physics, mass, charge, and energy. And so you alternate, when you build a model, you're alternating between a zero node, which conserves mass or charge, and a one node that conserves energy. But note that the sum of V's equals zero only contains V, nothing, no U's, no potentials, whereas the other one, the sum of U's only contains U, no V's. So to couple these, you've got to have a relationship between U and V, or between U and the quantity or the flux. And that's called a constitutive law, or a phenomenological relation, or a black box, if you like. It's capturing the physics of the material properties in a relationship between U and Q or V. And that's applicable to both energy storage and energy dissipation. So if you think about energy storage, you can either store energy mechanically in a spring, electrically as charge in a capacitor, or chemically as a solute in a solution. And each of those is a relationship between the potential, the movement, if you like, the stretch or the charge or the solute amount, and the corresponding potential that generates, either a mechanical potential, electrical potential in volts, or a chemical potential. And those relationships between U and Q are the material relationships and contain the material constant. So they have the elasticity or the electrical capacitance or the thermodynamic coefficient from Boltzmann's equation. And if you have dynamic processes, then you can also store energy in the inertia of mass or in the changing magnetic field of an electromagnetic system. Likewise, if you're looking at energy conversion across those domains, mechanical, electrical, chemical, you have a relationship between U and V, which could be a viscosity, an Ohm's law resistance, or in the case of a chemical solute, it's a Marsland-Redondo formula, gives you the flux through a reaction, which is dissipating heat. And in bond graph forms, we represent that either with what we call a single port resistance or dissipation device for mechanical, electrical, or in the case of chemical, it's a two-port, because you've got to separately account for the potentials either side of the reaction. And the chemical side of this is very much the work of Kachelski at Caltech, who added to the framework developed by Painter at MIT. So these are the constitutive laws of material constants that we need in addition to the conservation laws of physics. So let's give an example. Here's a simple protein model, a glucose transporter, the simplest possible one for a membrane transporter, GLUT2, facilitated diffusion inside the membrane. So here's the protein in the membrane, which is facilitating the transport of glucose, initially outward facing. So we have a state that we represent. The state of that protein is represented by that quantity Q, E naught representing the enzyme in the outward facing configuration. And then we bind a solute, which in this case is glucose. So you've got to be able to represent both the binding process, which is here, to then create an enzyme that's now got bound glucose, and then a reaction that represents the fact that that enzyme, that protein in the membrane, is switching from an outward facing to an inward facing position, and then releasing that glucose. So there's a reaction for the transition, and then there's another reaction for the release of glucose, and then there's a reaction for the return to the outward facing configuration. So you need basically 4 reactions to capture the physiological function of this transporter. And in bond graph forms, what that would be is this is a zero node where you're balancing mass. So the rate of change of storage of the state of the enzyme is the flux coming in minus the flux going out. So that's the zero node mass balance together with the storage associated with the bound glucose to the enzyme. And then you have a reaction which is representing at these one nodes is where energy is conserved, and they're giving rise to a very simple stoichiometric relation where the forward potential for this reaction is just the sum of the potentials from these two quantities, and then the flux through the reaction is given by the Marsland-Dedonda formula shown here. So you just do that for all reactions, and you get a model that you can then also simplify by using what's called the Briggs-Haldane assumption, where you assume that this is spinning at a constant rate. It's a steady state representation of the enzymes, but there's flux going through this system. And then when you fit data, Low and Walmsley data for the flux of glucose through the membrane in both directions, or rather changing the concentrations on both sides, we can get very good fits to the data, and we can also get very good fits to the measurements with the steady state approximation for the Briggs-Haldane equation. That's all described in this paper here. So if we now said, let's apply that to a larger class of transporters, to all the SLC transporters, it's of the order of 500 genes, 66 gene families, all the common transporters that move things across membranes when there's not ATP involved. And you can divide that into families of 1, 2, 3, or 4 transporters, mostly in the 2 or 3 range in either direction. So they include all the very familiar transport processes that you need to describe cellular function. And then we can just build bond graph models for all of those. And I've also got some of the ATPase pumps here too, the sodium, potassium, and the Circa pump. So they're all built in exactly that same fashion of just representing the key physiological reactions for the state transitions of the enzyme as it binds the ligands and then gives them up again. And then because some of these are electrogenic, for example, the sodium, well, quite a few of them are electrogenic, where you've got to keep track of charge movement as well as mass transfer. So the bond graph framework allows you to make sure you're always satisfying charge conservation as well. So here's an example. If you had a cell where in this case, an epithelial cell, and we're dealing with transport between the epithelial duct and the capillaries, as Bernard was mentioning, we'd have SLC, the SGLT1 transporter bringing sodium and glucose in, the GLUT2 taking glucose into the blood, and then the sodium-potassium exchanger, making sure that you're keeping the sodium concentration low inside the cell and moving potassium in as well, which is then driven by the ATP generated by the glucose. It also, because you're hydrolyzing ATP, you've got water flux also entering the picture, and this is just representing the aerobic metabolism for turning the glucose into regenerate ATP from ADP. And then because some of these are electrogenic, the SGLT1 is moving 2 sodium charges in, so it's electrogenic. So Faraday's constant here appears as you deal with the charge movement associated with that molar flux. And then likewise, NKE is moving 3 sodiums out for 2 potassiums in, so there's a charge movement as well. So the bond graph framework allows you to deal with the mass flow of all these different chemical species and the electrogenic effects. But also, because water is being transported, there's mechanical effects here too, because you're altering the pressure volume behavior of the cell in a soft-celled organism. So you've got to be able to handle the mechanics associated with the elasticity of the membrane and that water flux. So it's a good example of moving energy between chemical energy in the form of either the solute concentrations or the glucose bonds, and you're generating electrical storage in the membrane. It's producing heat, of course, and you're swapping energy between different chemical species, different chemical forms of storage, but also mechanical energy. So all forms of energy in any physiological process, you'll always get energy being transferred constantly between these various forms. So here's a more complex example where just using the bond graph framework is building up all the components for the cardiac myocyte for looking at both mass movement, charge movement, and energy conservation. And these models can be generated pretty automatically with once you've got annotation of all the individual protein components. And then they run very fast. They're much faster than real time in terms of running the models in software. Here's another one, which is pH control and neurons, which I won't go into. And then this is then going up a scale where you're now saying if you have models of particular cellular function for endothelial cells and epithelial cells, then you'd want to combine them into a tissue level, a functional tissue unit, as Bernard talked about, where we've got both transport through the cells into interstitial space, but also paracellular transport. And you're dealing with both the flux of the plasma solvent and the solutes in blood, but also likewise in the epithelial ducts. And sometimes you've got to also think about the fluid lining in those epithelial ducts. So the overall scheme with bond graphs is to represent everything is handled through bond graphs in terms of protein models that can then be simplified by using Briggs-Haldane, for example, building up functional cell units, which deal with all the processes within cells, being able to build those cells, such as the epithelial cell that I mentioned, or cardiac cell, from those intracellular processes, and then building up, as Bernard's described, the functional tissue units that depend on that knowledge graph of, well, the knowledge graph is used at all stages, but being able to combine the epianatomy framework with the physics-based bond graph framework. And then when you move up to organs, very often you've got to, and also at the cell level, you've got to deal with 3D diffusive processes, for example, and at the organ level there's spatial variation that needs to be handled through partial differential equations. So bond graphs is a zero-dimensional version of a more general theory called Port Hamiltonians, which we use for the partial differential equations of organ function. And then when you get to the whole body again for homeostatic processes, we can use the bond graph framework again. And then all the bond graph models are then converted into CellML, because we've been working for many years with the standards community to make sure that the models can be encoded, serialized, and standardized form, and made freely available on the model repositories, CellML, for example. And there's about 1,500 models currently in that repository, but we're in the process of converting many of them into bond graph form just to make sure that they satisfy the physics you're always satisfying mass, charge, and energy balance, which is especially important when you run the models for long time frames. You can't afford to have drift, so you've got to be really making sure that you're satisfying the physics. And then finally, here's a group of sources for the software that I've been talking about, and a journal which we publish called the Physiome Journal, which allows you to get additional credit for the curation and documentation, etc., of the models. A CellML site gives you access to many of the CellML encoded models. And I'll stop there, but leave that one up. Thanks very much.
Thank you, Mackenzie.
Thank you so much. I want to give the audience an opportunity to ask any questions. So if you have any questions that you would like to ask, please either raise your hand and I can take you off of mute, or you can ask in the chat or question and answer. Julie?
Thank you both so much. That was fantastic. I guess while we're waiting for other questions to chime in, I'll ask a few myself. So Bernard, for your part of the talk, I'm curious, how well conserved are these sort of systems that you show between organisms? You showed some examples in human. Has this been done in other species as well? And do you find major similarities and differences there?
Starting with the premise that the devil is in the detail. And the second premise is that, of course, a lot of SPARC data is in practice coming from animal models rather than human. On the other hand, the driving use case to develop electroceuticals is ultimately aimed towards the FDA level type of human type of modeling. So there will be absolutely some differences between organisms. For example, there are some organisms that do not have certain autonomic reflexes. Some species do not vomit, for example, or some species do not have a gallbladder. But also based on 20 years experience teaching physiology, I think it would be useful to focus on the commonalities and on the patterns that constantly recur in terms of multicellular organisms, and absolutely being mindful that there will be some key differences, because ultimately these organisms have evolved distinctly because they are fulfilling distinct ecological niches. So of course there will be differences. Nevertheless, you still need to have exchange between blood and cerebrospinal fluid. You still need to have exchange between the gut lumen and the lymph. So what can we sort of pare down to the most basic topological representation so that we have at last a stable topological network of compartments that need to ferry substances from one part to another? And really, it's being able to at last track meaningfully the exchange of solutes from one major thoroughfare to another, and then use the same topological scaffold to overlay the species-specific gene products that are brokering transport, that are pumping, that are serving as the receptors or the enzymes that are detoxifying a drug. So in a sense, while your question is very urgent, we're still very far from being able to answer that question in detail, because we still have to get the basics right. So the hope is that, based upon the lessons learned with the autonomic nervous system, which forces us to think about how the brain represents the rest of the body, and in particular how the primordial parts of the brain represent physiological function in terms of tracking pressures, stretch, and concentrations. Can we at least get those basics right and see how much actual processes we can classify and organize using this very basic framework? Epianatomy, though it looks very complex, is still an achingly basic framework when it comes to tissue compartments. There's so much nuance, but at least it's a start.
Questions?
While we're waiting, I'll ask one other. So I remember sometime in the past when I was looking at SPARC's website, I came across something about how certain collaborative programs are looking at things like non-Western medicine, right? Like acupuncture and other
Oh, right.
techniques and things like that. I'm curious how much of these types of maps and mathematical modelings within those maps are applicable to those sorts of um, understanding how those types of treatments could be working through the autonomic system.
Peter, maybe you might want to bring in some of the work that the ABI is focusing on, particularly in terms of looking at fascia representation, but also to create 3D body scaffolds to map these acupoints onto the same sort of framework. But it's useful to add that, again, coming to fascia, which is a fairly underrepresented network of connective tissue around the body, so little is known as a kind of gatekeeper for the transfer of tissue fluid, in particular across organs, but also as very clever cladding around nerves and blood vessels. That it seems that a lot of the effect of acupuncture, but again, I'm not coming from a point of a position of strength here, in that I'm not very familiar with the nuances of these set of techniques. But it seems that one avenue by which acupuncture might have an effect is really to influence fascia environment, and through that have a knock-on effect on the nerves and blood vessels that are passing through that fascia. But I'm not in a position of strength to comment eloquently about the effect of these treatments. And maybe Peter, perhaps you can add it. Go ahead, sorry.
Maybe I could just switch gears a little bit here, because there's a question from Grace Ping asking about, can I envisage AI assisting in the coding of physics-based models in bone graft form? And the answer to that, I think, is yes. I think, and we are looking at that, the huge advantage of having created CellML and having a lot of the physiological models, bone graft included, in a standardized form that's freely available for the web searchers to discover, is that it knows all about, if you dump a CellML model into Gemini 2.5, for example, it'll tell you exactly what the model's doing. It'll tell you, you can even ask it questions on how to extend the model, and it'll do that for you. You can combine models, and it's because you're operating in a very constrained environment of having standardized the way you express those models. And I think as we develop the bone graft models more comprehensively, where you're also making sure that the physics is always constrained, because you're always satisfying conservation of mass, conservation of charge, and conservation of energy, I think that'll be very useful in constraining the way that generative AI can put these models together, because it's got you can constrain it to make sure it is satisfying the laws of physics as it puts them together with the knowledge framework that Bernard's been talking about. So yes, Grace, I think there is a very good possibility of AI helping here to build these complex models.
So much for joining us today. We greatly appreciate having you here with us. With that being said, our next session will be at the end of October, so we hope to see you soon. And thank you again, Bernard and Peter.
Thank you. All the best.
Thank you.
Thank you.